Hi, my name is Kathleen Bostick. I am a graduating senior biology major from Spelman College, and I am really excited today to talk to you about soil microbiomes. This specific investigation has to do with the metro Atlanta area, and I am particularly interested in speaking about the taxonomic analysis and inhibitory interactions among bacteria. Now, what exactly consists of the soil microbiome? Well, it's a sublayer of the entire soil that's interacting with the roots, and it's made up of a large community of bacteria, fungi, archaea, viruses, and protists. Now, in this community, a lot of different relationships are established in order to allow everyone to thrive and really coexist with each other. An example of this is mutualism, where both parties benefit from the same thing. An example of this can be found in bacteria and fungi, where they get yummy, delicious sugars, and in exchange, they take care of the plant. Now, understanding plant health and sustainability is absolutely essential to allowing us to be able to understand how we can further improve crop development for humanity as we continue to persist in a world where climate change is continuing to affect our lives. So that brings me to my overall research questions and objectives. When you isolate a certain bacteria from soil, that is known as an isolate. I'm particularly interested in understanding how that isolate actually plays a significant role in the soil microbiome. I have two study sites presented in this investigation. One is the Spelman Victory Gardens, and the Spelman Victory Gardens dates back all the way to 1904. It has such a rich history. So understanding how these bacterial isolates actually play a role in a soil microbiome that is that old is really important to us understanding what kind of interactions it plays as a whole. Comparing this with the Oxford Gardens, which is a garden found at Emory University that was recently established in 2011, that soil microbiome is relatively new. So understanding how these isolates play a role in different soil microbiomes, given the context of multiple timelines, is really interesting in helping us understand how that overall affects soil microbiome. This brings me to my overall objectives, and the number one objective is really being able to identify the bacterial isolates. After identifying this, I want to retrieve the metagenomic data that was being used to examine the specific species found in the roots versus soil. This will give me an understanding of how the geographical region is affected in the context of the same area. All of this together will help me further seek knowledge on how these isolates truly do play a role in the previous interactions of the soil microbiome, as well as be able to predict the role that they may possibly play in either garden. Now, as I said before, the study sites in this investigation are Spelman Gardens as well as Oxford Gardens at Emory University. And Spelman Gardens dates all the way back to 1904, so it's very, very rich in history. Compared to Oxford College, this garden at Emory University really only dates back to 2011. And only in 2014, it was reached at its full capacity of being used. So being able to use this helps us understand the context and the history in multiple timelines. As you can see here, we have some wonderful pictures of the sampling that took place. As it pertains to the metagenomic data, these samples were taken in the spring of 2022, as well as the bacterial inhibition analysis was obtained in fall of 2022. In this investigation, we have multiple parts. The first part is the metagenomic data. This allows us to understand the bacterial isolates that are being classified. The bacterial inhibition allows us to understand how these bacteria is inhibiting other bacteria in the soil microbiome, which gives us a further understanding of what kind of role it plays in the soil microbiome as a whole. Now, once these samples were collected from multiple places, which gives us variety of a lot of species diversity, we removed rocks, roots, and debris to allow only the purest of substance possible. This was then stored at 4 degrees, and what we have here are the true methods and materials of how this investigation took place. The first was isolating the bacteria from the soil. Following this was gram staining. Gram staining is a really important step in understanding if this bacteria is gram positive or gram negative. This determination really highly affects the protocol that is used in order to undergo DNA extraction in quantity. Following this step, library preparation and nanopore sequencing through Galaxy is conducted in order to classify the specific bacteria. As you can see here, the metagenomic analysis through Galaxy is a flow chart. The number one takeaway from this is that you start with a group of data at step 1. Step 2, you do a trigonomatic, and this is done through FASTQ. And in this data set, it's cleaned up. So all you have is accurate data that is reliable. From the FASTQ set, you go to step 3 where you undergo FASTQC. And then you go through step 4 for Kraken 2 that allows us to classify this data and be able to determine what kind of bacterial isolate has been found. As you can see here through our taxonomic data is the alpha diversity. This allows us to understand what kind of diversity is really present in these bacterial isolates. The number one takeaway from here is in the observed, you can see that the rhizosphere has a significant amount of alpha diversity when compared to the roots of the Spellman gardens. However, when we see in the Shannon diversity, we see that the overall rhizosphere has a more significant amount of alpha diversity when compared to the roots. Now, further investigating this a little bit deeper, it's not just enough to understand the alpha diversity. We want to understand the relative abundance. This allows us to understand what kind of bacteria do we really see at the phylum level. What you can see here is a large amount of pink and a large amount of orange. Orange represents the actinobacteria found, and pink represents the proteobacteria found. Further looking at this, in actinobacteria, a very interesting thing was found. As you can see here in the diversity at the genus level, we see a large amount of pink, and this pink represents Streptomyces. Streptomyces is really important for producing almost 80% of the antibiotics that exist. So further looking at Streptomyces in terms of at the species level, the diversity, we see a very interesting upregulation in the roots of the Oxford Gardens. As you can see here in this column of root 01, there is this beautiful green here that is slightly different from the large overwhelming amount of green that represents the genera of species. This particular lighter green represents Grisio rubinosis. And this particular bacteria is really interesting because it actually originates from Russia. And this particular isolate that was identified is really important for antibiotics as well as antibiotic and anti-cancerous agents. Now, a second most common bacterial isolate that was found was Streptomyces ceylonensis. And this particular bacteria originates from Korea, and it produces a very specific enzyme that is really important for energy metabolism. What we also found was biosynthetic gene clusters, also known as BCGs. The number 2 that we found was melanin and ectoine. Ectoine is really important for osmotic stress, and as you know, yes, plants do have melanin, and it does protect them. So why is this information relevant, and how does it actually impact us as human beings as it pertains to this investigation? What we found here is that the specific isolate that I was responsible for understanding and analyzing was from the Oxford Garden. It is represented as O1 number 10. What you can see here in this graph is that it does inhibit one specific isolate, specifically JW number 2. However, it is also being inhibited by 2 other bacterial isolates, IS number 2 and O1 number 09. So it's very evident that not only does the bacterial isolate that I was investigating play a role in the soil microbiome, but it also inhibits. Not only does it inhibit, but it's also being inhibited by. So in conclusion, what we found most majorly within this entire investigation is that the most highest abundant of bacteria that was found was actinobacteria and proteobacteria. Actinobacteria had a high amount of Streptomyces that was specifically under the genus Grisio rubinosis. And this particular bacteria was really interesting in terms of understanding the antibiotic contribution that it pertains to the soil microbiome. Now, out of the 50 total colonies isolated by the lab that I am proudly part of, my one isolate pertained to a lot of really interesting inhibitory interactions. Now, this is all really wonderful. However, the next steps that I plan on taking is really understanding how these interactions are present among a larger number of isolates. And even further, understanding Pseudomonas specifically because it's very similar to Streptomyces. Understanding how both of these bacterial interacts will help us understand more of the microbiome. I'd love to acknowledge my lab as well as the Bio 487 class for helping me along this investigation. I would love to show you my references and answer any questions that you may have. Thank you.